So today we are going to continue on. We already talked about DNA replication. Now we're going to talk about cell replications. We're going to start with mitosis. We're going to practice it today, tomorrow. Probably have a very short quiz about it tomorrow. Probably won't be more than like 10 questions or something like that. And then we're going to cover meiosis on Thursday. Friday we'll practice it. I think we'll have a small quiz on that, and then potentially a test on it either Monday or Tuesday. This is a very short unit. So today we're going to talk about mitosis. And the first thing I want you to do is with your partner, and this is question number 1 for that activating activity. Which of these do you think has the most amount of chromosomes, and be ready to justify your answer? Raise of hands. How many thinks the dog has the most amount of chromosomes? No one. Okay. How many think that the human has the most amount of chromosomes? 1, 2, 3, 4, 5, 6. Raise your hands high if you think the humans have the most chromosomes.
I think they do.
So we had 1, 2. Hold your hands up so I can actually count. 1, 2, 3, 4, 5, 6, 7, 8, 9.
8. No.
How many think that the adder's tongue fern has the most? 1, 2, 3, 4, 5.
Did you not? I said they got. He said it wasn't, guys. Why are you raising your hand? You're wrong.
Okay. I'll save this for later.
It's the dog.
No,
It's
just
the
tell
dog.
us now.
It's the dog.
Okay.
Either way.
So the one thing I will say, I'm actually going to go over this. Chromosomes are not a measure of complexity. So just saying.
It's the leaf.
So we are going to. I will go over this here in a little bit. The next one. I'm going to label these as 1, 2, 3, 4, and 5. How do you think that these would go in order? Write the number in order from 1 to 5 that you think this would go in order. Ashlyn, what order do we think that it's in?
1, 3, 2, 4, 5.
So we're going that one, that one, that one. Can you justify, why do you think that it's in this order?
I don't know, just by looking at the picture, so when it goes from 1 to 3, it goes from, like the yellow dots, they slowly move apart, but they're not completely.
Okay.
And 2 and 4. 4 looks like it's just being cut in half, but separate. It's like slowly separating, so that's why I said that, and then 5 is the last one.
Okay. So you notice that the yellow things are moving apart. In this one, they're even farther apart. In this one, they're starting to separate out. And then this one, they're kind of separated. That's a good idea. Yeah, so this is what we're going to be talking about in this unit. We're going to be talking about chromosomes. We're going to be talking about all that fun stuff. And we're going to be talking about how cells replicate. So this is going to be our process of mitosis.
I'm close.
So we are going to go more in depth with it.
1, 2, 3, 4, 5.
What?
You weren't even close to me.
Yeah, so in order, it would, I can't move it. In order, Ashlyn had it right. But if you remember, back when we talked about cells in Unit 5, we said that all living things are made up of cells, all things are the basic units of life, and all cells come from pre-existing cells. So we're going to be talking about cells and focusing on this one right here. All cells come from pre-existing cells. So we're going to be talking about how cells create new cells. Yes.
Yeah, how did the first cell come to be?
Lots of evolution.
Okay.
So this is going to align with our LEQ. How do cells reproduce, and how does this reproduction manifest itself in unicellular and multicellular organisms? We're going to be covering this one. So we are going to be talking about kind of how cells reproduce and how we make more cells in our bodies. So the first thing that we have, if we remember prokaryotes and eukaryotes, hey, guess what? It's coming back.
No.
So prokaryotes. What do prokaryotes have?
1 cell.
One cell and
Produced by
Binary fission, they divide by
Do they have a nucleus or no nucleus? No nucleus. Eukaryotes then have a nucleus. So we are again differentiating this. Prokaryotic cells, in this case, are going to reproduce asexually. Asexually means that there's no egg and sperm, so they are going to just kind of divide. In this case, they are going to be dividing by binary fission. So that is just the separation of the cell into two cells. Eukaryotic cells, much more complex, need to do a lot more, so we are going to call this mitosis. So we are going to be focusing on mitosis more so than we are going to be binary fission. But we have to not only replicate all of our DNA, replicate all of the nucleus and that kind of thing, we also have to figure out how we're going to move all of the organelles, because there are a whole lot of different organelles that we've talked about that are filling up this cell. So we have to be able to divide them out in order to get to fully functioning cells. The other thing that we are going to look at is the difference, at least we're going to talk about it briefly, the difference between a plant cell and an animal cell. So we said they had a bunch of different organelles. For example, plant cells had chloroplasts, they have cell walls, all of that fun stuff. Animal cells have the centrosomes, which we'll talk about those here later. But in this case, the way they divide is also going to be different, because plant cells have that big cell wall. It makes it a lot harder for them to divide. Animal cells can just kind of pinch off, since they're kind of gummy. I like to think of them as like gushers.
Ew.
I like the nerd
I don't
guy.
like that
Mhm.
feeling.
But they can just create what's called a cleavage furrow. So this is the kind of divide where they kind of pinch off and separate. And plant cells, on the other hand, since they have that cell wall, they can't just pinch off like the plant cells can. So as a result, whenever they divide, they build this kind of layer between the cells, and this is called the cell plate. So the cell plate is going to just appear in plant cells. It's kind of like if you cut yourself and you get a scab, it's not going to completely protect yourself from the outside world, but it's going to hold it over long enough for the skin to heal that spot. Same thing is happening here. It's kind of like making a scab to divide the cells until we can completely build the cell wall between the two of them. Any questions about this? So like I said, animal cells have a cleavage furrow. Plant cells have a cell plate. So, for our cells, we are going to, because scientists love to classify things, we are going to classify our cells into two main categories. Most of this that we're going to talk about is animal cells. This is happening in plant cells as well, though. We have two different classifications. We have gametes and we have somatic cells. Gametes are, if you remember, your egg and your sperm. It's going to be reproductive cells. These are going to be something called haploid. So we're going to talk about haploid versus diploid here in a bit. But your gametes are going to be haploid. And they are going to reproduce through something called meiosis. Meiosis is what we're going to talk about the second half of the week. Somatic cells, on the other hand, are all of your body cells. They're going to be diploid, and they're going to reproduce through mitosis. So you can see on the bottom, you have your germ cells, your gamete cells. They're going to reproduce to create 4 haploid cells through the process of meiosis. Somatic cells are going to reproduce through mitosis and create 2 diploid cells. With this, what I want you to do, with your partner, discuss what you think the difference is between being diploid and being haploid. So you don't have to write this down anywhere, but discuss with your partner what it means to be diploid versus haploid.
All right. So,
What do you think the difference is between diploid and haploid?
Half of it?
Okay, so if we break it down, haploid versus diploid, you see the N versus the 2N.
Yeah.
So this one could be half of this one.
Oh, yes.
Okay, you're not entirely off. Anyone else have any other ideas?
So you have to solve for n.
I understand.
So
Oh,
you put
2n equals
you put 2n equal
Put
to
2n
n, right?
equals n
And
divided
if you get n by itself, so you divide by 2.
Oh,
No, wait. Let me take it.
No. Okay. Yes, it would be half.
So you have to factor n out.
If we're doing math, it would be 2n equals n, n equals.
2 plus 1.
Oh, yeah.
That would be, if you were doing math, that's how it would work out. However, in the case of diploid and haploid, all of our cells are going to have 2 copies. They are referred to as diploid. Di means 2. Haploid then means half. So the way I usually remember it, haploid, HA, half, HA. So this is going to have half of the cells, or half of the chromosomes, that a normal cell would have. So, our haploid, like I said, half the genetic material from the parent cell. So if a cell has 10 chromosomes, a gamete, in this case, a germ cell, would have half of that. So they're haploid. They have half of the genetic material. So if we have 46 chromosomes, each one's going to get 23. Diploid means 2n, or diploid or 2n means that it's going to have the same amount of chromosomes as the parent. So somatic cells, you can see, turn into 2 diploid cells, meaning they have 2 copies of each chromosome. That is going to be very important when we actually talk about chromosomes, but that won't be until next week when we talk about mutations. So a lot of this is leading up to mutations. A lot of this is more prep work. We have diploid, same amount as parent. Haploid, half as much as parent. Okay. So what we're going to do with this is we're going to practice this. Welcome to math class.
Don't like that.
Hold on. So we are going to go through the first one, and then I'm going to have you practice with the rest of them. So, the first one. And this is also going to answer the first question that I had. So, a diploid human cell has 46 chromosomes. How many chromosomes would a haploid human cell have? So 46 divided by 2.
23.
It's going to give us 23 chromosomes. So this is not hard math. So, Matthew, if a haploid ibex, anybody know what an ibex is? It's a type of goat, yes. Has 30 chromosomes. Matthew, how many would a diploid cell have? 60. If we're starting with half, 30 times 2 equals 60 for a diploid cell. Starting to make sense?
Yeah.
The adder's tongue fern has 1,440 chromosomes.
1,000.
Emily. Guys. Emily. It'd have 720.
You said it wasn't the plan.
I said it was. I didn't answer what it was. I
Oh,
said
you did.
I
You
said
lied.
I said that it was not a measure of complexity.
No, whenever we were over here, we were like, it's the plan. You were like, it's not the plan.
I didn't say that.
Oh, you did.
But
Come on. You heard it.
whenever we looked at the, actually, we'll go over the rest of these and then say, Kyle, fruit flies, Drosophila melanogaster, has 2 chromosomes in its unfertilized eggs. How many chromosomes would a somatic cell have? 4. So a unfertilized egg means that it is a gamete, so it's going to be a sex cell. So 2 times 2 is 4. Fruit flies only have 4 chromosomes. Surprisingly. That's why they're so easy to work with for genetics. Cade. Domestic dogs have 78 chromosomes in their muscle cells. How many chromosomes would its gametes have?
39.
So 78 divided by 2 would be 39. And last but not least, Ashlyn. Each pollen grain has 10 chromosomes. How many chromosomes would a fertilized diploid egg or an ear have? 20. So we're starting with pollen grains. Pollen grains are the reproductive, they're the eggs and, well, in this case, the sperm of corn. So 10 chromosomes times 2 equals 20 chromosomes. So going back to the beginning, whenever I said which has the most amount of chromosomes, the fern had the most amount of chromosomes.
Yay.
Yes. Surprisingly, this is one of the organisms with the most chromosomes on Earth. It's a very simple, it's called the adder's tongue fern. I forget where they grow. However, you notice dogs have 78. We only have 46. So it's a matter of how our instructions are organized. It's not a measure of complexity. Because usually we think of ourselves as very, very, very complex, but there's a lot of instructions needed for the other organisms.
Do we know what plant or animal has the least amount of chromosomes?
A snail.
I honestly do not remember. I can look it up though. So hold up. Guys, the organism with the least amount of chromosomes is a male Australian ant, Myrmecia pelosula, which has 1 chromosome per cell. So, does this make sense? Like I said, haploid has half, diploid has the normal amount. So now, what we are going to do with the last few minutes, we are going to go through mitosis and meiosis. We're going to start with mitosis today. We're going to work on it a little bit more tomorrow. And then, like I said, meiosis will be Thursday. So, our cell cycle, only 10% of our actual cell cycle is devoted to mitosis. It's a very short and quick process. So you can see in this cycle, in this calendar, we have G1, which is a growth phase, S phase, which is the DNA replication we just talked about yesterday, and we have G2, which is another growth and preparation phase. So all 5 of these stages that we're going to talk about are jammed into this last section. Yes. And it seems like there's a lot happening, but it's a very quick and easy process. But first, we need to talk about chromosomes. So chromosomes. You'll see on your next page there is a diagram of a chromosome. We're going to label them. So the first thing to note, each individual arm, so we have one right here and one right here. These are called chromatids. So a chromatid. So each individual one is a chromatid. Yes. Yes. So yes, each one is a chromatid. The thing holding them together here in the middle is something called the centromere. So centro is central. So this is the central point on the chromosome. Sometimes it's found in different spots. So for certain chromosomes, it'll be way up at the top. Sometimes it'll be in the middle. Sometimes it'll be at the bottom. Yes. So we're not going to worry about the arms. However, the other thing to note that is not on your paper, your genes, and this is something we'll want to write off kind of to the side, your genes are found on these arms. So for example, right there is some sort of gene. So you'll want to make note that the genes are found on the arms, so something like this. And these are going to be wound up pieces of DNA. So this is a lot of DNA wrapped around a bunch of proteins, and this is a very compact and easily portable version of your DNA. The last thing that you need to notice, we have our chromatids. These are going to be, if we have two chromosomes that look exactly the same, have the same centromere, same arms and legs, these are going to be homologous. So homologous, Logous chromosomes, which means that they are going to be the same. Typically, they have information stored on the same spot. So if we look at gene A and gene B, they are both found on the same spot. Yes. So these are individually, they are sister chromatids. Together, they form homologous chromosomes. That's where we're going to end it for today. Like I said, in our entire cell cycle, let's look at this like a calendar, like if you have different things you have to do over the course of the month, especially, actually, let's look like this was a calendar of December. How much school do we have in December?
31 days. 15 days.
We have a very short amount of time in December
It's
because,
15 days.
yeah, because half of that is cut off for Christmas and holiday break. So kind of like that, only a small portion of the calendar, in this case, is based or is used for actual cell division. Ashlyn, come on. You know better.
Just take it.
You know better at this point.
Here we go.
So, only part of the calendar is devoted to our cell cycle. The rest of it's devoted to growth. You see up here, it says cell growth. Over here, DNA replication, the thing we talked about on Monday. So we need to make a bunch of copies of our DNA in this S, or the synthesis phase. Synthesis, if you remember correct, back when we talked about dehydration synthesis. Synthesis is to make, so we're going to make copies here. And then this G2 phase is going to be more growth and more preparation for when we go into this mitosis. Mitosis is going to be a little bit of a stressful process for the cell, because you are literally taking all of your body parts and you're splitting them in half. Half goes to one side, half goes to the other. But before we talk about the actual splitting of the cell, we need to talk about how we're going to organize our chromosomes. So chromosomes are the condensed version of DNA. They are wrapped tightly around these little proteins called histones. And these histones are going to kind of coil it up. Actually, I think I have a string here. Actually, better yet. So imagine my lanyard is our DNA. The histones are going to wrap the DNA around it and kind of coil it up so it's easier to store. So these histones are then going to be wrapped up and they're going to be turned into our chromosomes. So we start out at the most stretched out, the most laid out part with our DNA double helix, and we end up with our most tightly coiled chromosomes. So when we look at our single chromosome, so this is on, I think, your third page, you can see we have a whole bunch of parts. Some of these we're not going to look at, some of them we're going to look at more in depth. For example, each one of these sides, normally, yes, they look kind of like an X. They aren't actually X's, weirdly enough. One side of this is called a chromatid.
Yes.
Yeah.
So these two, then, kind of like how we've been using female analogies for all of these are called sister chromatids. And they come together to form a chromosome, and they meet at the centromere. The centromere, centro is central, so they're going to meet at this central point on the chromosome. Like I said yesterday, sometimes the centrosome is not dead set in the middle, so you'll get some chromosomes that look like this, some that look normal in the middle, and some that have really long sections with the centrosome kind of lower on it. So it's not always dead set in the middle, but it is going to be where the two chromosomes meet up. Yes, Noah?
You know, like when you're like graphing, like tangent. It's kind of like that a little
Yeah, kind of. So like if you're graphing all of those, you can see where it hits the line. The point where it hits the line is like the center zone. And then the other thing that, like I said, is not on your paper, anywhere.
So that is 2 chromosomes in one pair.
Yes, this is 2 chromatids to make 1 chromosome.
Oh.
Yes. We're going to model this later, so it'll give you a better idea. And then anywhere on this chromosome, for example, if I were to look right here, I could find genes. Genes are going to be the things that code for our different characteristics. We'll focus a lot on this when we get to genetics. Genetics is a lot of fun. But our genes are going to code for your hair color, your eye color, different diseases, different personality traits. All of those are going to be coded in our genes. The other part we need to look at when we are looking at pairs of them, like I said, if we have 1 chromosome, it is made of 2 chromatids. If we have 2 pairs of chromosomes, they are called homologous. So let's break this down. So homo, where have we heard homo before?
Homo sapiens.
Okay, Homo sapiens, sure. Anywhere else? Homosexual? Yes, I know. I'm going to dive into this again. So we said way back when that heterosexual means you're attracted to the opposite sex. So what is homosexual? Same sex. So in this case, homolog or homologous means that they are the same structures. So, for example, if you got 2 homologous chromosomes, you got 1 from your mom, 1 from your dad, because we have the same 46, they're going to be the same. So like chromosome 1 on your mom is going to look the same as chromosome 1 on your dad. The only one that's going to look a little different that we'll talk about a little bit later are the actual X and Y chromosomes, the ones that determine whether you're male or female, because they look different. But otherwise, homologous chromosomes are going to look the same. So let's go to here. Let's do it a little more organized. So chromosomes are made of what? Strands of DNA. Okay, let's just. And that DNA is made of what? Genes. Okay.
Yeah, shoot. My bad.
And even though it's not up here, what code for these genes? Not
No,
XY.
X, Y is chromosomes.
I mean, traits, these will code for traits.
Is it the
ATCG, which are the
Apple tree card garage.
what is that type of molecule? Nucleic acid, or well, monomer of a nucleic acid. What is the monomer of a nucleic acid?
It would depend on my face.
Nucleotides. So our chromosomes are wrapped up pieces of DNA. The DNA is made of genes, which is coded by nucleotides. Those nucleotides code for our genes. Those genes indicate what traits we are going to have. That is a type of chromosome.
Y and Y, X and Y and.
So any questions about this? Guys in the back. Any questions?
Sorry, sir.
Pay attention.
I'm sorry, sir.
Pay attention.
Nope, stop.
Yes, sir.
There we go. So now we're actually going to get into mitosis. So mitosis, there's going to be a very easy way to remember this that I will explain after we go through the individual steps. But what you are going to do, we have our little chart here. You are going to jot down the step what's happening in the step, and I want you to draw it so that you have a visual representation of what's actually happening. So the first one that this is going to be a layout. So we start out in our interphase. Our interphase was that G1, the S, and the G2 phase, so that big majority of the calendar. And then we're going to jump into, that was good, our actual cell division. So we start out with a stage called prophase. From there we go from prophase, I'm going to jump over prometaphase because it's just kind of a middle step. We're going to move from prophase over to metaphase. The way I usually remember that, meta is middle. So it's going to jump to metaphase. Metaphase is going to go to anaphase. Anaphase to telophase. And then finally, they're going to divide off in a process called cytokinesis. So we're going to go through all of these in depth. So the first one, prophase. Prophase is our beginning of cell division.
Yes.
Yes. So there's a big chart for this one. So prophase. Things to note, if you were to look at a microscope and see a cell, how could you tell that it's in prophase? The first thing to note is that the nuclear membrane is starting to disappear. It's starting to kind of dissolve. The other thing to note is that your chromosomes are starting to form. They are appearing condensed. If we saw a cell that has a big dark nucleus, that will be interphase. If it is dark, that means there's a lot of chromatin. The DNA is spread out, relaxed like you on the couch. And it's all laid out so that it can be easily accessed. Whenever we go into this, it starts to get condensed and wrapped up so that it can move to the new cell. The other thing to note are these little yellow things up here. Anyone think they remember what these are? Yes. These are the centrioles or centrosomes. So the centrosomes, we said, in animal cells were important for cell division. It's only found in animal cells. What?
How do you want us to draw this?
So we have our cell.
You just want to like
Straws on board.
Yeah. Something like that. So the main things to look for, the nucleus is starting to dissolve, so you could draw that as like a dotted line. The chromosomes are becoming apparent, and the centrosomes are starting to move. So you can see they have these long arms here, and these are going to be really important when we get to our next step. But the centrosomes, we said, were only found in animal cells, and they are important for cell division, so that's what we're going to use them for in this case. So plant cells don't have these things, just our animal cells. So our next stage then is going to be called metaphase. So metaphase, like I said, meta is like the middle. So in this stage, we have our chromosomes have been fully removed from the nucleus. The nucleus is no longer there. And what you can see is they are lined up in the middle of the cell. We call this the metaphase plate. This is going to be kind of the dead center of the cell. They're going to line up in order, kind of like if I just had you guys stand in a line up front here. So we have very condensed chromosomes. They're going to line up here on this plate. And the spindle fibers, these little arms coming out of our centrosomes, are going to attach to each one of our chromosomes and just hold them there. And this is going to lead into how we are going to split everything up. Next one is anaphase. So anaphase is when we start using our centrosome, our spindle fibers, and we are going to start pulling the cell apart, or not the cell apart, but pulling the chromosomes apart. So the chromosomes, they're like besties. They don't want to be pulled apart. So this is going to take a lot of energy on the centrosomes part, but they're going to start pulling these chromosomes apart and pulling them to the different poles, the different sides of the cell. Kind of like how we have the north and south pole on the Earth. These ones are going to start being pulled to the poles. From there, we move on to telophase. So this looks kind of like a doubled version of prophase. In this case, what is happening? The chromosomes are now at the poles. They are at the different sides of the cell. They're going to become more diffuse. They're going to start, now that they're separated, they're not hunting for their sister chromatid. They're off on their own. We are going to reform we're going to start to reform our actual cell or nuclear membrane. And at this point, it's mostly a division of the cytoplasm and the cell membrane. So you can see over here we have this pinching, kind of like if you were to pull two pieces of like Play-Doh or two pieces of slime apart, it's going to pinch in the middle. And we said yesterday, this is called a cleavage furrow. So this pinching is called the cleavage furrow. That is where the cell is going to split in half. Anyone need more time with this? We've got one more step. And that is cytokinesis.
There's no explanation.
There is. So in this one, the
Here, I'll let you copy it.
So in this one, cytokinesis, kinesis is to move. So telekinesis is to move with your mind. So cytokinesis is to move, in this case, the cell. Cyto is the cell. So cytokinesis, we are splitting apart into two new cells. These are our daughter cells. Each cell has its own DNA, and it has a copy of each organelle that it will need to survive. So all of the organelles, even though they weren't pictured in these images, also got moved to each of our cells. So we now have two fully functioning daughter cells.
And then you can divide eventually, right?
Yes. So then the cycle repeats. We get interphase. We get our growth 1, synthesis, growth 2, and then these two will divide. And then the 4 that they produce will divide. And the 16 that they produce will divide. And at that point, you would now be an embryo. Or something along those lines. So all of the cells in your body, all of your skin cells, all of your muscle cells, all of your nerve cells, all of those were created by mitosis. Any questions about the steps of cell division? Now there is, hold up, there is a very easy way to remember this. Because remembering prophase, metaphase, anaphase, telophase, cytokinesis is a little much.
Yeah.
Anaphase, telophase, cytokinesis.
Oh, no.
So there's a very, guys, hey. So there's a very easy acronym to remember this. What are the first 4 letters? PMAT. Everyone say it? PMAT. PMAT. P-M-A-T. Prophase, metaphase, anaphase, telophase. And then you just have to remember cytokinesis. So you could say PMATIC, But PMAT. That is how I have remembered this for many, many, many years. So in order, looking back, we have our interface. Guys, I should not hear side conversations. We have our start. We have the nucleus is starting to dissolve. It's starting to disappear. So this would be prophase. Metaphase, it's lining up at the middle. Anaphase, they are starting to pull towards the poles. And the last one, telophase. Because we are starting to reform our nuclei, and the cell is slowly pulling apart. Any questions about mitosis? When we go over meiosis, all of these steps are going to show up again.